for the kind invitation. It's a pleasure and honor to be together with you today. And indeed, I would like to share with you some of our work, which is focused on adaptive characteristics within innate immune responses. And the first question is, of course, why would we be interested in such a subject? Because memory responses are especially described to take place in adaptive immune cells, and they are very important for memory T and B cells, and that is how we build vaccines. But we started to be interested in that when we worked with mycobacteria and with BCG some almost 15 years ago now. And we had seen some strange processes happening in our patients that were vaccinated with BCG, in which we were seeing heterologous responses after vaccination. And we asked ourselves, is it possible that BCG might have also effects outside of protection against mycobacteria? And when we look back into the literature, we have seen, and I have to say that before those studies, I was not aware of these old epidemiological studies showing that when BCG was introduced in various populations around the world, it induced a decrease in mortality, which was stronger than that can be explained by protection against tuberculosis alone. This is an old study done in Sweden by Carl Näslund, who was introducing the BCG vaccination in Sweden. This is a slide actually that Peter Aby from Denmark kindly agreed that I can use, and you can see here that after the introduction of BCG in children in Sweden, there was a decrease from 11% to 4% in the total mortality of the children. Of course, this is before the antibiotics, when unfortunately a lot of children died. And you can see also that the TB deaths were very low, so they could not explain the complete protection given by BCG. Well, they observed that the neonatal sepsis and non-tuberculosis infectious diseases were also decreased. And Carl Näslund concluded that there must be a non-specific type of immunity that protects these children. And we started to be interested in this, and this type of epidemiological observation have been made many times around the world when BCG was introduced in the population. Peter Aby and Christina Stabel-Ben did also a number of clinical trials actually in West Africa showing the same thing. And we started to be interested to understand, well, what precisely might be the immunological mechanism behind that? And can we also repeat this observation in a controlled manner in the human population? So first of all, we did a study in which individuals received either placebo or BCG, and then we administered yellow fever vaccine 1 month later as a model for an infection, because of course it's not easy to infect somebody who's healthy, but when we do yellow fever vaccination, we actually induce a very mild infection with the yellow fever vaccine virus, which is an attenuated virus that we can measure with a PCR in the circulation of the vaccinated individuals. And you can observe here the viremia in the circulation on day 3, 5, and 7. And in the beginning and at the end, there were very few viruses in the circulation. In the beginning, it was too early. In the end, we eliminated the virus, obviously. But on day 5, most of the individuals in the study had viremia in the circulation. And as you can observe, the BCG vaccinated individuals had significantly lower levels of viremia, meaning that although this is a very mild infection, but still BCG was able to protect against the non-specific viral infection that has nothing to do with mycobacteria. We also tested the yellow fever antibody titers because we wanted to know whether BCG decrease in the viremia would not result in a significant decrease in the antibody titers, but that was not the case. On day 90 after the yellow fever vaccination, the antibody titers were similar between the two groups. So with this in mind, we ask ourselves, well, how is it possible that this happens? What could be the mechanism? And of course, now the induction of a specific immune response by memory T and B cells is unlikely to explain this observation because that is antigen dependent. And there are no common antigens between yellow fever vaccine and mycobacteria. But then we asked ourselves, would it be possible that maybe the function of innate immune cells, which are non-specific, might be also modulated by BCG vaccination in an antigen agnostic manner? And we purified monocytes from these individuals, and then we stimulated them either with Mycobacterium tuberculosis, but also with heterologous pathogens such as Staph aureus or Candida albicans. And what we observed actually is that these individuals displayed an increased function of their monocytes independently of the type of stimulus that we used. So their monocytes were more responsive. So their innate immune cells were more responsive. And then we asked ourselves, what could be the molecular mechanism between this increased responsiveness of innate immune cells after vaccination? And these data are all published. So I'm just summarizing basically a couple of years of experiments, basically, is that what we have observed is that in a naive innate immune cell, it can be a monocyte or a macrophage, in the regions of the genome where the proteins are made, which are important for host defense, there is no gene transcription. The chromatin is in a closed conformation because transcription factors do not need to bind to the promoters to initiate gene transcription. And we change this when we activate the cells, basically, and this is well known. Histones are methylated, acetylated, phosphorylated, and so on. Chromatin changes its architecture. It becomes more loosely packaged. The transcription factors can bind to the promoters, initiate gene transcription, and so on. Now, when we eliminate the infection, we always thought we lose these chemical changes, and then the transcription stops and we go back to normal. And this is more or less correct, but not completely, because most of these epigenetic changes, these chemical changes in the histones are lost, but not all of them. And some of them persist, especially methylation of the enhancers remain present, and they bookmark the genes which have been activated in the first place. And when the cell is re-stimulated, this bookmarking allows the process to go on quicker and more efficient. It's like having a book and now having a bookmark and knowing exactly the page where you need to read it, so you go directly to that page. And that results in the end that during the second infection, the responsiveness of this innate immune cells is more efficient, increasing the immune response and protecting better the host. Now, once again, very important, this process is not dependent on a certain antigen. It is antigen agnostic, and second infection can be with something different. It just needs to activate the same genes, and the same genes need to be needed for the host defense against the second pathogen. Otherwise, it doesn't help. Now, we asked ourselves at that moment, okay, if this bookmarking takes place, which genes are bookmarked? And actually, we assessed 3 of the open chromatin histone modifications, monomethylation of lysine 4 of histone 3, so H3K4 monomethylation, H3K4 trimethylation, and H3K27 acetylation. And you see here, these were monocytes purified, exposed for 24 hours to the training stimuli. Then we washed away the training stimulus. We let the cell rest for 6 days, and then we assess the chromatin accessibility. And then we look at these histone marks, and you see that this last column in each of these colors actually are the genetic loci which have an enrichment in these open chromatin marks. And then we perform pathway analysis of these genes which were bookmarked, and we observed, well, lots of genes that are important for innate immune responses: interleukins, inflammatory genes, toll-like receptors, integrins, and so on and so forth, which is logical. This is what is happening basically in the beginning. But at the same time, we observed also a lot of genes that were important for cellular metabolism, oxidative phosphorylation, glycolysis, amino acids metabolism, and so on. And then we asked ourselves, why would the cell do that? And first of all, yes, it is also about energy. The cells needs more energy when it's activated, and probably this is the reason why glycolysis is increased. But there are also other mechanisms that are at play. And what we observed is the following, is that there is an integration between the immunological signaling induced by the stimulation of the cell with changes in cellular metabolism and thereafter long-term epigenetic processes. What do I mean by that? What we observe actually is that stimulation of certain pattern recognition receptors, in this case it's beta-glucan depicted in this picture, but the same is also with peptidoglycans from BCG, from mycobacteria. They induce intracellular signaling such as PI3, ACT, mTOR pathway that induces changes in cellular metabolism, increased glycolysis, important as an energy source. But at the same time, there are changes in the TCA cycle. The TCA cycle acts now anabolically instead of catabolically. Citrate is extracted from the TCA cycle, forms acetyl-CoA, and acetyl-CoA is a donor of acetyl groups for histone acetyltransferases, so the histones can be acetylated. But at the same time, it enters the cholesterol synthesis pathways, and one of these metabolites in the cholesterol synthesis pathway, mevalonate, is secreted and provides an amplification loop of these processes by stimulating IGF-1 receptors. At the same time, the TCA cycle is replenished through amino acid metabolism, especially glutaminolysis, and leads to accumulation of fumarate. And fumarate is an inhibitor of KDM5 histone demethylases. And because we inhibit these histone demethylations, the methylation of the histone is allowed to persist. So basically, this process allows the methyl group to bookmark the enhancers on the histones. Now, this is very nice, and we show that this is happening in circulating monocytes and also in vitro. But there is a problem with this whole process, and the problem is that the monocytes in the circulation that we have studied in all these experiments, they are there only for 1 to maybe 3 days. But we have seen these changes after 14 days. We have seen changes in these monocytes at the epigenetic level and functional level on day 90, so 3 months later, even one year later. So we ask ourselves, well, how is that possible? Well, the only potential explanation for that would be that similar type of changes would take place after vaccination, also at the level of hematopoietic stem cells, which actually give birth to all these monocytes in the circulation. So in a follow-up study, what we did, we asked a group of volunteers to get the BCG vaccines and to donate not only blood, but also bone marrow aspirate. And we looked at their blood. Their blood was inducing this increased function of the monocytes, of their innate immune cells. But then we looked at hematopoietic stem cells. And we purified the hematopoietic stem cells from the bone marrow, and we performed RNA sequencing and ATAC sequencing, so chromatin accessibility and RNA sequencing. But even at RNA sequencing alone, and we had also some placebo vaccinated individuals to be sure that the process of taking the bone marrow is not the one which influences the gene transcription, and it was not the case. But on 90 days after the BCG vaccination, so at the moment that we could not find any mycobacteria anymore in our volunteers, we see that the transcriptional program of the hematopoietic stem cells is significantly changed, with an increase in myelopoiesis and granulopoiesis, bias and more activated cells. And that was true also at the level of the chromatin accessibility. And what was very interesting was we were seeing a quite strong bias towards granulopoiesis. And we never have studied granulocytes because we thought, well, even more than monocytes. Granulocytes are end-stage differentiated cells we won't see any difference there, basically. But now seeing this very strong bias towards granulopoiesis, we thought, would it be possible that granulocytes in the circulation are also changed after BCG vaccination? And actually, we purified in another study the granulocytes from BCG vaccinated individuals. Then we stimulate those macrophages with MTB, with Candida albicans, LPS, PMA, and as you can see here, the release of IL-8 of elastase was significantly higher in the neutrophils after BCG vaccination. We also have looked at the capacity of these neutrophils to kill microorganisms such as Candida albicans, which again has nothing to do with mycobacteria, and we have observed that the killing capacity significantly increased in these neutrophils after BCG vaccination. If we looked at various mechanisms that are responsible for the killing capacity, as I showed you previously, the elastase was increased. Also, the ROS production, reactive oxygen species, was increased. The net formation, on the other hand, was completely normal. Nothing changed in the net formation after the BCG vaccination. Very interestingly, even with the rather small number of individuals that we had in our study, we observed that actually histone marks of open chromatin in the neutrophils, such as H3K4 trimethylation, in pro-inflammatory genes such as IL-1 beta, IL-6, and TNF, significantly associate with the amount of colonies of Candida albicans. The more open chromatin, the more the capacity of the cells to respond to stimulation, the less amount of colonies of Candida albicans after co-incubation. So there was an association between the epigenetic program of these neutrophils and their capacity to kill Candida albicans. Now, all in all these studies allows us to propose that certain vaccines, and I talked about BCG vaccine, but there are other colleagues have shown beautiful work with other types of vaccines, and I'll go in a couple of minutes also a little bit about that. But some vaccines are able to induce signals, and the question is which signals, and probably there is a combination of the release of bacterial factors, but also endogenous mediators that travel to the bone marrow level, and they induce a reprogramming of the hematopoietic stem and progenitor cells in the bone marrow, which induce thereafter epigenetic, transcriptional, and functional changes in the innate immune cells that allow these innate immune cells to respond stronger to a re-stimulation and to improve the protection of the host. Now, is this being done in the same way by everyone? Well, the simple answer is no. There is a lot of variation, as with everything. If we have different lengths and different colors of hair, why wouldn't we have different immune responses as well? So we were interested in one of our more recent studies in trying to understand what are the factors that influence the way in which healthy volunteers respond to the BCG vaccination. And we knew also from the previous studies that there are people who respond extremely well with the induction of these long-term increased responsiveness of the innate immune cells, what we call trained immunity after BCG vaccination. And there are individuals who don't, who actually respond poorly to that. So for that, we recruited a cohort of 300 healthy volunteers. We vaccinated all of them with BCG, and we collected blood on day 0 and day 90. And then we assessed basically their immune function and the variability of the induction of trained immunity. You can see here that BCG induces changes in cytokine production capacity. You see a lot of things that are improved. And then we looked at different factors, different host factors, immune cell frequencies. How do they influence these responses? And there were some effects, but not very strong, I have to say, of age and sex and BMI and so on. There were also some cells which are strongly associated with the immune responses, especially monocytes, as one would expect. What was very interesting to observe, on the other hand, is there was a pretty clear seasonality effect with higher responses in terms of trained immunity in the winter. And I'll come back, why do we think that that is the case? And then we assessed basically responders and non-responders in terms of chromatin accessibility. And we observed something very interestingly. First of all, epigenetic profile of monocytes are associated with the trained immunity responses, with the responsiveness of the monocytes after the vaccination. However, we found actually very little changes in the epigenetic profile of the T cells to be associated with the adaptive specific immune responses with interferon gamma induced by Mycobacterium tuberculosis, which indicates that epigenetic processes are probably, an epigenetic variability is probably more important for innate immune responses, trained immunity responses, than for adaptive classical adaptive memory, which might not be so surprising. But we observed also a second thing, and what we observed basically, and this is in panel C, is that individuals who had open chromatin from the beginning, before they got BCG, they do not increase strongly their cytokine production capacity after BCG. They already have a high cytokine production capacity in the beginning, and they do not increase it even more. Basically is, if you have an open door, you cannot open it even more. Whereas the people who had a good response to BCG were the people who in the beginning had poor cytokine responses and a closed chromatin profile. Meaning that indeed, if the door is closed, then you can open it and the cytokine responses, the immune responses are better. So there was a very clear association between the epigenetic profile before the vaccination and the way that people respond to the vaccine. And again, we also try to see whether we can predict the trained immunity responses and also the adaptive specific interferon gamma capacity based on the epigenetic profile of the people. And we can do a reasonably good job, actually. You can see here ROC curves to predict the trained immunity responses. But based on the epigenetic profiles of the cells in the circulation of the healthy volunteers, we could not predict the strength of the interferon gamma production induced by mycobacteria, so the adaptive immune responses, which are most likely an antigen process, antigen-dependent process, and variability is dependent on other factors. Okay, now we have learned a little bit about the variability of the immune responses within the population. But what about the variability in the responses within an individual, between the cells of an individual? So basically, we see this induction of trained immunity in monocytes, for example, of an individual, but do all the monocytes respond the same? So for that, we performed another study in which we purified monocytes. We exposed them actually with various stimuli that induce trained immunity, BCG, beta-glucan, also metabolic stimuli, oxidized LDL and uric acid crystals. Anyway, we trained the cells for 1 day. Then we let the cells rest. We wash the stimulus. We rest the cells for 5 days. And then we re-stimulate them with LPS. And 4 hours later, we collect the cells and we perform a single-cell RNA sequencing to assess whether all the monocytes, in our case, responded similarly with an increased responsiveness. And to make a long story short, that was not the case. There were some approximately 25–30% of the monocytes which responded perfectly fine to LPS stimulation, but not more after they were exposed to BCG. Okay, then we had another 25% approximately of the population that responded with more production of chemokines, especially CXCL9, 10, and 11 that has been previously shown to be strongly upregulated by BCG vaccination. And we call these monocytes producing chemokines, basically. They were trained, but especially for production of chemokines. And then we had thereafter approximately half of the population which responded with increased production of chemokines, but especially with increased production of pro-inflammatory cytokines, such as IL-1 beta, IL-6, TNF. We call them monocytes producing chemokines and inflammatory cytokines. So there is a heterogeneity, thus, also in an individual who is responding otherwise well, but not all his cells respond the same. Then we extracted the transcriptional programs from these two types of cells, monocytes producing chemokines or producing chemokines and inflammatory cytokines, so MC and MCI transcriptional program. And we looked in various databases with transcriptional data to see whether we can identify them. Well, we identify them, for example, very easily and quickly, very strongly in inflammatory bowel disease. So that is very interesting to look further. But then we looked also at COVID-19 because at the time we were doing these studies, we were in full pandemic. The pandemic had started. And what we have observed, and these are not our data, let's say in the RNA-seq data are not our data, but are single-cell RNA-seq data by a German consortium published in Cell in 2020. But what we have observed that if we look at MC and MCI transcriptional programs, we identify it very easily in individuals with mild COVID, but not in individuals with...
Hello, Charles.
So individuals with severe COVID were not able to mount a proper monocyte response, apparently. So we thought, well, combined with all the epidemiological data, would it be possible that we have the following concept, that there are individuals which have high innate immune responses from the beginning when they encounter SARS-CoV-2, when they are on the street or they go to a concert or whatever, they respond very good. Because they respond with a high innate immune response, they inhibit the growth of the virus. They will have low viremia in the end, low systemic inflammation, low symptoms, and survival. But there are maybe some other individuals who have low innate immune responses in the beginning, and they allow the virus to grow when they don't even know that they are infected, but they allowed multiplication of the virus much easier, that grows a lot. They have a high viremia, which induces an inefficient systemic hyperinflammation, high severity of the disease, and unfortunately, some people will die. So we ask ourselves, would it be possible to boost these innate immune responses like the children do with BCG? So maybe we can protect against BCG. Protect against infections also in the other risk groups in adult individuals. And first we did a study, the ACTIVATE study, which was done before the pandemic, 2018-19, in which elderly people who were in the hospital for other reasons, when they went home after recovery, they were randomized to receive either placebo or BCG, and we followed them up for 1 year for the number of infections, and we observed that indeed they had approximately 40% less infections than the non-vaccinated individuals. So we thought, okay, this is very interesting and potentially important also for the pandemic. We were at the beginning of the pandemic, so we thought, let's try to protect the people also against SARS-CoV-2, and that was the moment that we did not have yet the specific vaccines that we have now. And we performed the BCG Prime study in 6,000 individuals in the Netherlands. These are also elderly. We gave them BCG or placebo, but unfortunately, we were not able to see any difference in the total number of infection. Okay, so this is clear answer. We cannot protect. We cannot see the total number of infection being changed. But how about the immune responses and the severity of the disease? So we assessed thereafter immune responses in the individuals who got placebo or BCG vaccination and then COVID-19. And what we have observed is that those individuals have better cellular immune responses and also better humoral immune responses. As you can observe here, cytokine production capacity and also IgG and for S protein, N protein, and RBD region. So we thought, okay, what about then the severity of the disease? We fortunately didn't have enough numbers in the study of people getting into the hospital or dying. But when we looked at COVID-19 related admissions, we have observed approximately 15% less admissions in the BCG group. We also have looked at influenza infections, which was diagnosed actually on clinical grounds, so influenza-like infection, I have to say. Interestingly, so not SARS-CoV-2, but non-SARS influenza-like infection was again 50% less. And I will not have time to go into detail, but we have performed thereafter also animal studies observing that BCG protects much better against influenza than against SARS-CoV-2. And there is a question, why is that happening? But anyway, thereafter, we also looked at the mortality, and this is BCG prime study. And we had 13 deaths in the BCG group and 18 in the placebo group, approximately 30% less in the BCG group. Fortunately, well, not many, but still it seemed that there is a certain protection, but we could not reach statistical significance. But then we asked our colleagues who did other studies with BCG during the pandemic to provide us with their mortality data. And when we performed summary statistics together with Peter Abi and Christina Stabel-Ben, who also did their own studies, we observed approximately 40% reduction of the mortality in the BCG vaccinated individuals, as it has been seen previously with other infections. Okay, this is about BCG. Now the question would be, is BCG the only vaccine that can induce these non-specific heterologous innate immune responses? And the answer is no. There is a beautiful study, and there are more studies coming up from Bali Pulendran, who looked at seasonal influenza vaccine, and he performed assessment in non-adjuvanted and adjuvanted influenza vaccine, and he observed a very strong and potent increase in antiviral trained immunity responses if he used an AS03 adjuvanted influenza vaccine, which showed that actually also some influenza vaccines can induce similar long-term responses as BCG is doing. And ourselves, we were also interested in measles-containing vaccines, because measles was a second vaccine that has been described in a lot of epidemiological studies that is protecting against, non-specifically against infections in children, especially in Africa, but also in other studies. And this is a systematic review published by a WHO SAGE group now 8 years ago, showing that measles-containing vaccines actually had a clear protective effects in the children. And we were interested to understand, are measles-containing vaccines, in our case MMR that we have studied, do they have similar type of immunological effects as BCG? And for that, we vaccinated healthy volunteers with MMR, and then first we collected blood before vaccination, and 1 month later we looked first at cell counts. They were not different, but with BCG also we don't observe many differences in cell type numbers. But then we looked at the function of the monocytes, and we expected to see precisely the same as the BCG, more improved function of the myeloid cells. And to our surprise and disappointment, we were not able to see anything. There were absolutely no difference in the responsiveness of myeloid cells before or after MMR vaccination. And then we started to scratch our heads and thinking, well, what is happening? What is MMR actually doing? So we had no clue, but we had the PBMCs actually before and after BCG vaccination, and then we decided, let's do a single cell RNA sequencing to see which cells have the most changed transcriptional program. Maybe we have to study those cells. And to our surprise, the cells with the most genes changed in the MMR vaccination group were the gamma delta T cells. And gamma delta T cell is a special population, subpopulation of T cells. It's stimulated, generally antigen independent, a sort of innate-like type of lymphocyte. So that was very interesting. And then we thought maybe it's the gamma delta T cell function that is modified by MMR. And indeed, when we assess the function of the gamma delta T cells in terms of cytokine production capacity that was increased. We have looked also at metabolic processes in gamma delta T cells. We observed a shift from glucose use towards towards a higher fatty oxidation and the capacity to sustain higher level of gene translation. And we are trying now to understand how is this inducing the protection. But in any case, what we have observed is the gamma delta T cells in the case of the MMR vaccination are much more changed, both transcriptionally and functionally, compared with myeloid cells. So apparently different vaccines can induce different trained immunity programs also in different type of cells. So all in all, what we would like to point out is that certain vaccines can combine induction of memory characteristics in both adaptive immune cells, TMB cells. Very important. I would never say in contrast. This is probably more important basically for protection against the specific pathogen, the specific TMB cells. But at the same time, certain vaccines can induce also adaptive characteristics in innate immune cells, in myeloid cells, in neutrophils, even in gamma delta T cells. And that can be important also for protection. And then they can also give antigen non-specific protection, so antigen independent protection. And in the future, we might consider actually combining vaccines that induce a proper adaptive immune memory with those containing inducers of trained immunity to obtain a broader protection against more pathogens. So what I wanted to share with you today was the fact that we see after infections, and I didn't have time to discuss more, but also infections can do that, but also vaccination, they can induce epigenetic reprogramming of innate immune cells that can be at different levels. I talked especially about histone modifications, but DNA methylation changes take place, also modulation of non-coding RNAs such as microRNAs and low-no coding RNAs are different and then can lead to changes in the function of the cells. There could be also at certain levels tolerance programs which are important, especially at the mucosal level. These are induced by the microbiome, for example, in the gut and so on. But at the systemic level, they can also induce trained immunity programs, also at the level of the hematopoietic stem and progenitor cells. And that can be very important for innate immune maturation, non-specific protection by vaccines. On the other hand, and I don't have time now to go into that, if these programs are inappropriately activated, wrong place, wrong time, they can lead to immune paralysis, the tolerance programs, for example, in sepsis or in cancer, or they can lead to hyperinflammation in tissue, atherosclerosis, neurodegeneration, and so on, the trained immunity programs. And finally, of course, I would like to thank all the colleagues who collaborate with us and are helping us in the studies. And also to mention that if you are interested in this subject, there will be the 6th International Symposium on Trained Immunity in Dresden in Germany in November. Thank you very much.
Thanks a lot for your presentation. There were many questions in the chat. And maybe one of the first question was what is magic in a vaccine that will result in trained immunity? And is it something that is restricted to live vaccines? Whether you had the chance to test other vaccine platform, viral vectors, mRNA, LNP vaccines, of course, do they imprint innate cells also? And whether it's the same way as BCG, for instance.
Yes. So this is an excellent question, very broad question at the same time. Well, in my opinion, there is nothing very special into this actually, because epigenetic changes after an initial gene transcription activation are very old processes. These are very ancient processes that take place. I do think that, well, they happen all the time that we activate gene transcription. It is impossible to activate gene transcription without epigenetic modification. And often there are some epigenetic scars that persist. Sometimes they persist and they are useless or even deleterious, but also millions of years of evolution have taken care that in innate immune cell, we use these processes for building an improved immune response thereafter. And this is happening after many infections, and people have shown this happening. That doesn't mean that is always beneficial. For example, in contrast, there are many occasions when there is deleterious long-term effects. For example, after sepsis, we know that there are long-term immunosuppressive effects that are induced by sepsis, leading to increased susceptibility to infections, to increase also atherosclerosis development, and so on. So some of these epigenetic changes are not necessarily beneficial. And the difference between, well, why is sepsis inducing something else than BCG, for example, I think is the type of the stimulus, the strength of the stimulus. We know when we overwhelm a cell with stimulation, that is also happening with adaptive immunity. If you overwhelm a T cell with too much antigen, it becomes tolerant to that. And that's the same with an innate immune cell. If you stimulate it too strong, there are certain metabolic pathways that are activated in the cells, starts to accumulate NAD+, activates sirtuins, which deacetylates everything, all the histones in the cells and so on, induces tolerance, and it's inducing long-term immunosuppression, basically. So it depends a lot on the balance. What type of immune cells are activated? How much do they activate it, and through which type of receptors, and what are the immunological signaling and metabolic processes that are induced. And that determines actually the trained immunity program that is activated. So I'm going now to the second part of the question. Are all these stimuli or vaccine activating similarly the cells? Absolutely not. And trained immunity is not one program of trained immunity. It's a concept, basically, and there could be many trained immunity programs. We also see that the BCG program of trained immunity is very different from that induced by MMR, and that is also different from that induced by influenza, which is a strong antiviral influenza vaccine, which is a strong antiviral program that has been shown beautifully by balipulendron. And not all the vaccines can do that. Some of them actually can induce tolerance. We have shown that, for example, DTP induces tolerance. But some of them, maybe many of them do that. Many of the live microorganisms can do that because they are mimicking actually an actual mild infection. And a mild infection is more often beneficial because it boosts the immune response and usually results in trained immunity, whereas a very severe one can induce actually tolerance and immunosuppression. Do only live vaccines induce that? Likely not, because we see now that AS01 and AS03 containing vaccines, so adjuvants, also induce certain programs of trained immunity. And then we can debate, are they better or not, and how much protection do they give? But at least there are some indications that, for example, AS01 containing Shingrix vaccines seem to be protective against severe COVID-19. And at least in the study of balipulendron, the AS03 containing influenza vaccine was able to induce infection with other viruses in vitro. So I think that more than only live attenuated vaccines can do that, but they will induce different type of programs, and they could be qualitatively different, whether they protect against the same type of pathogens or not, and some of them even inhibiting the immune responses that can also happen. Also with the new vaccine, it has been shown, balipulentron actually have shown also with the new messenger RNA vaccines, trained immunity effects. And there was a group from Ireland, from Trinity College, who showed also trained immunity effects of the AstraZeneca vaccine.
Okay, thanks. Another question related to the impact of the route of administration of vaccine, whether it's only the vaccine doing the trick, or also the route of administration of this vaccine is key to induce trained immunity.
No, absolutely. So the route is also very important. For example, our mucosa are meant to become tolerant because we have microbiome there, we have all the microorganism. If the immune cells in the mucosa are continuously activated, we would all have inflamed mucosa. We would all have IBD and so on. So at the mucosa level, usually we get tolerance rather than training. For example, the skin is a very different immunological environment compared with a mucosa. So there we do see that it's induced training. We know from non-human primates and from mouse work that, for example, BCG works by far the best if given IV, probably because of the accessibility to the bone marrow. But of course, I'm afraid at least to give BCG IV, so I wouldn't do that. So it depends a lot, but of course we are busy and other people are busy actually to mimic the BCG, so to give it safely IV, for example, but without the side effects of a live microorganism, of course.
Thanks. And another question in the chat related to the benefit of trained cells on adaptive cells. In other words, how do trained immunity could impact the specific adaptive immunity that is what we aim to induce with a vaccine usually?
Yes, no, absolutely. I mean, there is a continuous interaction between innate and adaptive immune system, and that goes both ways. Goes from the innate to adaptive immune responses, and I didn't have time to go to that. But this is what we have always done. Adjuvanticity basically is a part of the trained immunity response, basically, because it's improving one of the functions of the innate immune cells, which is antigen presenting. So, the only reason why we didn't just call trained immunity, well, adjuvant immunity or whatever we want to call it, is purely because the name adjuvant tells us, well, it's just helping the T cells. Whereas, in fact, this long-term epigenetic reprogramming of the cells takes care also of a direct antimicrobial function independent of the T cells. But that being said, the improvement of the T cell responses is a crucial component why trained immunity appears in the first place, because it's improving also the adaptive immune responses. And in turn, actually, adaptive immune responses, and there are some very recent beautiful papers published. There are 2 papers in Nature Immunology beginning of this year showing that actually interferon gamma coming from the adaptive immune cells, from the T cells, also from NK cells, which are innate immune cells, but interferon gamma goes back to the innate immune cells, and there is a loop and a strengthening of the response between the trained immunity response and the adaptive immune memory, and they strengthen each other basically, one through the adjuvanticity, the other through amplification of trained immunity responses by gamma interferon.
Yeah, so you need both together.
Absolutely.
Mm.
Well, I mean, if I would say, well, trained immunity is so important, we don't need T cells and B cells, this is absolutely stupid to say, because we see what is happening with the children with the severe combined immunodeficiency. They don't have T and B cells that function. They basically die without a bone marrow transplantation. So T cells and B cells are crucial. On the other hand, we have the monomax syndrome in which people don't have monocytes and dendritic cells. Many of them also die of infections, and all of them would die if we wouldn't have antibiotic, and many of them also have to get bone marrow transplantation. So we absolutely need as mammals, basically, both of them.
Thanks. There was a question. I'm just reading it in the chat, but antiviral pathways are thought to be to downregulate pro-inflammatory cytokine pathways. and vice versa. And so it could seem counterintuitive in terms of subtypic trained immunity. And so what do you think about this balance between type 1 interferon and, let's say, TNF pro-inflammatory signaling?
That is true. There is a difference indeed, as you mentioned, and as the colleague in the chat was mentioning, there is also a difference between type 1 and type 2. Type 1 indeed inhibit inflammasomes, inhibit important components of the inflammation. That's why, for example, we do see that some of the vaccines, for example, the ASO3 influenza vaccine induces especially an antiviral response, not an inflammatory antibacterial response, let's say. So that is absolutely correct. It is different, though, from gamma interferon. And what I was mentioning by amplifying and strengthening is gamma. Type 2 interference is very well known. It is also inhibiting, actually, a little bit IL-1 beta, but is strongly strengthening TNF, tumor necrosis factor alpha, and the general antimicrobial function of the innate immune cell. So I think it's also a crucial difference between type 1 and type 2 interference. With type 1 especially inhibitory, but type 2 interference, gamma interferon, having also an important amplifying effects on innate immune cells and trained immunity. And that has been shown by several new studies.
There was a question regarding the quality of innate training and whether it is expected to protect more from viral infection or from any infections.
I think it depends once again of the type of trained immunity program being induced, and I do think in the end it depends on what kind of bookmarking are you doing in the first place. Because, for example, take again the example of BCG, there will be certain transcription factors which are activating, and they are depositing the epigenetic marks in certain places where they have affinity for, and the promoters and enhancers and so on. So depends on the transcription factors being activating in the first place. Whereas a very different vaccine activating very different transcription factors, they will bookmark basically different type of genes, leading to a very different profile of improved response. So depending on that, you will protect. For example, the influenza ASO3 showed very clear that trained immunity program, which was basically an antiviral trained immunity program, was very strongly geared towards protection against viruses.
Yeah.
Whereas BCG, for example, we see it now also, and again, we don't know exactly why. We are actually working to try to understand why. Also in animal models, very clear difference of BCG in protecting more strongly influenza than SARS-CoV-2. So we have to identify exactly why. It depends on the specificity of the first transcription factors, which epigenetic bookmarking is done, and how is the cell activated.
Another question asks how the original antigenic scene or humoral imprinting would influence trained immunity, like how trained immunity might depend on the presence of immune complexes or pre-existing antibodies. And so about the crosstalk between.
Yes, to say very simple, I don't think that it has been done, so I do not know. Nobody has looked at it. I think it would be interesting to assess, but as far as I know, it has not been done. So we don't know, basically.
Okay. And somebody asked what we could learn from vaccine-induced trained immunity about sepsis and the epigenetic program that result in a kind of paralysis or tolerance, about maybe how to erase some marks or whatever.
We and other people are busy indeed with this. There are conceptually very close processes. Trained immunity and tolerance induced by sepsis is just other genes that are up or down regulated, basically. We are trying to understand exactly what metabolic processes are important for that, and what epigenetic marks and what are the the readers and erasers and so on of those epigenetic marks. I do think that epigenetic modulation of the immune responses has still a lot to learn, and there are still a lot of possibilities actually in therapeutic targeting. This is taking place at this moment mostly in cancer, but I do think that also in inflammatory disease, autoimmune disease, where deleterious strain immunity programs are present, or in cancer and infectious diseases where the opposite tolerance programs are present, that we should learn to manipulate epigenetically the cells so in order to rebalance the immune response. And I think in the next years, a lot of work will be done in that direction.
Yeah, I think I've seen people talking about epigenetic adjuvant.
Yes, Bali
vaccine.
Pulendran actually is a very strong proposal of that. And in terms of rationally designing certain adjuvants that have certain epigenetic capacities, that's very interesting. But also pharmacological modulators in disease that act at epigenetic level in immune cells will be also very interesting in the future.
To test. And maybe one last question, if you are still okay. I think it's very interesting that you cannot push trained immunity, and there is a maximum, and maybe there is no risk of over training. What does it mean exactly? Does it imply that chronic stimulation, or if you live in an area where you are frequently exposed, at like a low age, you will reach a maximum of training in your innate compartment? I think it depends also how long these marks will stay.
Yeah, no, I mean, in the end, one cell has a certain capacity, and the capacity will be reached at a certain moment at maximum. So yes, a maximum can be reached. The problem is not, unfortunately, is that maximum does not mean that you cannot activate it too much. So yes, there is a maximum, but if you activate it in the wrong place, if the cell has the maximum, but in a certain place it should not have the maximum, or at a certain moment, the maximum should be reached, for example, when it's needed, when you have a severe infection, and for a short term you should do the maximum, but you shouldn't do it continuously and in the wrong place, and so on. So unfortunately, that's why deleterious strain immunity programs are still happening in inflammatory diseases. There are a lot of people working in atherosclerosis and train immunity responses in inflammatory bowel disease, for example, in gout and so on. So it can still happen, even if there is a maximum, yes, but the maximum should be used carefully, only when you need it.
Yeah, the balance is always hard to find.
Yes.
And somebody wonder whether epigenetic marks could differ between, let's say, mRNA LNP vaccines and like subunit adjuvanted vaccines, and whether there could be a link with these differential marks with persistence and or durability of response.
Well, I do think that there will be epigenetic differences between them because they will activate differently the cell. And at the moment that you activate differently the cell, the epigenetic scars that remain thereafter will be different as well. Whether that can explain differences in persistence and durability, we don't know. Actually, this should be done. But this is a very young field, and those studies have not been done yet. But I think it would be very interesting and important to understand it, yes.
I think we all agree that it's an amazing field, and really with a lot of impact in the way we think about vaccines and vaccine design, vaccine schedule, and so thanks a lot for opening our mind in that direction. Thanks.